I am one of two current faculty in the Division of MS and Neuroimmunology at UAB. My partner, Dr. Bashir, is on his way to Pakistan to visit family, which is something he doesn't get to do very often, so we wish him a speedy and safe travel. We're also excited this year to have a clinical fellow for the first time since I've been on faculty. Dr. Metter has joined us since the summertime, and it's been a very exciting time to see our clinics expanding and also to be working with some of the new therapeutic agents that have recently become available to us in the MS field, and also to look ahead to some of the new therapies that are coming our way in very short time. So since there is a lot going on in MS Therapeutics, I wanted to try and give an overview of exactly what is coming and what to expect. But I realize with all that information, it can be very challenging to distill it down to a 30-minute talk. So I'm going to try and give a brief survey and then talk a little bit about practical considerations of how we think about these drugs in relationship to one another. So first of all, just disclosures. I'm primarily funded through the Department of Veterans Affairs. I've also done some research funded by Biogen IDEC looking at symptomatic effects of their drugs, so nothing that really pertains to today's talk. And since I'm talking about therapeutics, including therapeutics that are not yet FDA approved, there's a long list of those drugs which are listed here. So I wish I could just come here and say, well, MS is an expanding field and there's more and more options, but in reality we live in a world where cost and comparative effectiveness have become increasingly important issues, especially when it comes to very expensive medications that are taken over decades, if not lifespans, like the multiple sclerosis disease-modifying agents. And so I just pulled a couple of quotes from the recent press. This is an article from a pharmaceutical news bulletin that talks about how some MS medications are being dropped from mail-order pharmacy formularies in the interest of cost-saving measures. And also here's a quote from the Washington Post. The article is mostly about the expense of long-term treatment of HIV and AIDS, but there were some quotes in there from neurologists as well concerned about the availability of multiple sclerosis drugs in some of the newer insurance programs that are trying to limit the various therapeutic options available to patients. And so we as MS neurologists really have to be mindful of the drugs we use and try to use the most effective drugs at the least expense. Because these pressures are not going anywhere. There's also some pressure from professional organizations such as the American Academy of Neurology, which issued some guidelines earlier this year to much controversy that try and encourage neurologists to think carefully about which MS patients they treat with agents such as the beta interferons and letermiracetate. I think those of us who see MS patients like to think that even though these drugs are studied in specific patient groups, that there are still individual cases where we make exceptions to those rules. And so we have to sort of very carefully thread the needle when we're deciding who is appropriate for treatment and so forth. So this is just a short overview of what I'm going to talk about. First of all, I just want to briefly review the drugs that we currently have available to us, including some of the newer oral agents. I'm going to speak again briefly about some of the agents that we'll likely be hearing about in the next 2 to 3 years. I'd like to spend a few minutes talking about how we integrate the information about these medications to make rational therapeutic decisions in our patient population. And then lastly, touch on some changing approaches to MS therapeutics that really go away from the traditional immune modulating effects to more neuroprotective approaches to treatment. So first of all, prior to 1991, this was the field of, these were the therapeutic options for disease modification in MS. And as you know, this has been a field that has become increasingly crowded over time as more and more agents have flooded the field, giving us not only lots of therapeutic options, but also lots of financial support for neurological publications, because I know you've seen all of these ads in the journals that you receive in your practices. Because I don't have the time to go into all of these drugs, I just created a table to summarize what the treatment field more or less looked like up until 2012. At that point in time, we had 3 preparations of beta interferon, as well as glutaramine acetate, all of which were administered by self-administered by injection at home. They had similar efficacy, reducing relapse rates by about 1/3 on average. And these drugs have all proven to be very safe to take over long periods of time. And so for many people, these are still the mainstay agents. And even many patients who are now aware of the newer drugs that are available have continued to stay on some of these drugs just because they do work and they're very safe and familiar to the MS patient community. Beginning in the mid-2000s, we had Natalizumab come along, which for the first time seemed to break through that relapse rate reduction rate of about 30% and get on up to about a 67% reduction in relapse rates, which really seemed to open up a new, or sort of raise the bar in terms of what we can hope to expect from the treatments we can offer our patients. But of course, the success of Natalizumab was somewhat tainted by the discovery that treatment with this drug in some patients seems to increase the risk of progressive multifocal leukoencephalopathy. And so because of that occurrence, this drug now is largely used as a second, third, or fourth line agent as opposed to a first line agent because of the concerns for this secondary side effect. Fingolimod was the first oral disease-modifying drug that became available in 2010 to much fanfare. And although it also appears to have efficacy that most likely exceeds that of the injectable medications, its use has been somewhat limited by the degree of monitoring that has to go in before patients start on the drug and then after patients continue on the drug, looking for things such as macular edema, skin cancers, and also monitoring for side effects like lymphopenia and hypertension. So this was what the field looked like up until about 2012. And since that time, we've had 2 additional agents become available, and I don't know how many of you are using these newer oral agents in your practices, but both of them offer something a little bit different than what was available before. So Teriflunomide, also known as Aubagio, is a once-daily oral medication that's a metabolite of leflunomide. And the way we think it works is it reduces activation, proliferation, and function of T and B cells through the inhibition of pyrimidine synthesis. It's convenient because it only has to be taken once a day. And while most patients tolerate the medication very well, there are some treatment-limiting side effects, including hair loss, which is mild to moderate in most cases, but can be severe in rare circumstances. You have to keep an eye out for hepatotoxicity. The drug does have a black box warning for teratogenicity, which has really limited its use in younger, especially female patients, although male patients are also advised to use contraception if they're taking this medication. And then some GI side effects, including diarrhea. When you look at the effectiveness of teriflunomide, it does have a modest effect on relapse reductions compared to placebo on the order of about 30%. So we don't have a comparison to all of the injectable medications, but it seems to be fairly similar in its scope of effect. When you look at disability progression, the results are fairly encouraging compared to placebo. Both the 7 and 14 milligram doses have a modest effect on reducing sustained disability progression. This just shows a comparison to subcutaneous interferon beta 1A or REBIF, which shows that the drugs are fairly equivalent when it comes to relapse rate reduction. The other new oral agent is Dimethyl Fumarate, otherwise known as BG12, otherwise known as Tecfidera. And this is again, operates on a completely novel pathway that hasn't previously been approached in multiple sclerosis. It activates this NRF2 pathway, which in addition to having some anti-inflammatory effects, Also is thought to have some, to mediate some protection against oxidative stress, so possibly there's some neuroprotective effects here as well, although that's not been clearly established. As opposed to the other oral drugs, this one is taken twice a day. And the most notable comment about its side effect profile are the things that seem to occur in the first 4 to 6 weeks of treatment. Patients will typically have episodes of flushing that can be quite intense. People will even attract the attention of others sometimes because they can turn very bright red just all of a sudden. It'll typically go on for 15 or 20 minutes before resolving on its own. That's not typically treatment limiting in most cases, but what can be treatment limiting are the occurrence of GI side effects. This happens in a small portion of the patients who take the drug, but when they occur, the pain, vomiting, nausea, and diarrhea can be quite severe. And we haven't quite figured out exactly how to ameliorate those GI symptoms to get people more or less over the hump of the initial first couple of months of therapy to keep them on it. And rarely the drug has been associated with lymphopenia, but in the clinical trial, the rate of lymphopenia was quite low. If you, one of my slides, or one of my charts didn't show up there, but there were 2 studies of dimethyl fumarate. One was a placebo, just a placebo-controlled trial. The other one included a placebo as well as an active comparator for the purposes of approval in the European market. And you can see compared to placebo that BG12 in both twice and thrice daily dosing reduced relapses on the order of about 45 to 50%, and slightly better than the comparator glatiramer acetate, which reduced relapses by about 30%, which is consistent with its previous clinical trial experience. Unlike the placebo, the initial trial, the benefit trial, in the active comparator study, there were no demonstrable benefits in reducing disability accumulation, but in the initial study, there was some effect on disability as well. It's notable to comment that this study was not powered to show a difference between Copaxone or glatiramer acetate and dimethyl fumarate. But despite that, it did show significant benefit over GA in annualized relapse rates and in some of the MRI metrics. So you'll see out there there's some comparison to Copaxone as this being a superior drug, and it may be better in some aspects, but it's not consistent across all metrics that we use to monitor disease activity and progression. Confusing the scene a little bit more is some of the newer agents that are now becoming available. The one that we're likely to hear about next is called Alemtuzumab, formerly known as Campath, soon to be known as Lemtrada. This is a anti-CD52 monoclonal antibody, which once administered, rapidly depletes both B cells and T cells in patients. It is given as an infusion over 5 days with the initial induction, and then it's redosed at 1 year and possibly a second year over 3 days. And that's really the extent of treatment with this particular agent. It's compared to all the other drugs we've used up until this time, it appears to be the most efficacious of all the drugs that have been studied for MS thus far. And because of its robust effects on efficacy, an FDA advisory panel recently recommended it be approved at the FDA's next meeting, which will come towards the end of this month. So we should hear about this drug and its approval status at the end of December. However, they did have some reservations about the clinical trial design, so it'll be interesting to see what the FDA ultimately recommends and if there's any additional recommendations made about further studies or about additional monitoring. One of the interesting things about this drug is that some of the phase 2 studies showed benefits on not only relapse rate reduction, but disability that were fairly profound and also quite sustained over many years. But these effects were primarily seen in patients who were treated at the earliest phase of the disease, which is a bit complicating because as a drug with potentially significant side effects, which I'll go into, it's likely not to be recommended as a first-line agent. So, Bart, some of these figures didn't show up. So there was a figure here that basically showed in comparison to beta interferon, I'm just pointing at blank space here, compared to beta interferon 1A, that it had substantial improvements in both relapse rate reduction and in disability accumulation. I have another figure later on, I hope it shows up, that shows what happens over prolonged periods of time in these patients. And of greatest concern as a adverse event for this drug is the occurrence of secondary autoimmunity. And no one really understands exactly why this secondary autoimmunity occurs, but it will be the main limiting safety factor for this drug once it becomes available. So patients who take Alemtuzumab are at high risk for developing autoimmune thyroid disease, which you can see here on the left. And there's some patients who will develop anti-thyroid antibodies but may not develop symptomatic hypo or hyperthyroidism. Also of concern, there were several cases of idiopathic thrombocytopenic purpura, including at least one fatality that occurred when a patient had an intracerebral hemorrhage. This was during one of the early phase 2 studies before it was recognized that this was a possible complication of the drug. And so one of the things that we'll have to do as neurologists who are using this medication is to keep an eye on patients once they've been dosed. So it'll be a little bit more similar to treating a cancer patient where you have to monitor for long-term side effects of the drug itself, perhaps more so than the MS disease activity. And then it can also cause some other more rare autoimmune conditions such as Goodpasture syndrome. So this is a drug that's not without its risks, but in patients who may have a very highly aggressive or severe case of multiple sclerosis, it may be a very appropriate therapy. One of the other drugs that will be forthcoming is ocrelizumab, which I think of as sort of the son of rituximab, whereas rituximab is a non-humanized monoclonal antibody. Ocrelizumab is the humanized anti-CD20 antibody that's currently in phase 3 trials for treatment of relapsing multiple sclerosis. And you can see in phase 2 studies, the drug compared to placebo has robust effects on reducing gadolinium-enhancing lesions. These are the placebo and interferon beta-1a groups. These are the two ocrelizumab groups. So gadolinium-enhancing lesions essentially disappeared in the phase 2 study. And if you look at relapse rate reductions, you can see that compared to placebo, that the ocrelizumab has quite a robust effect. Another drug that we're looking forward to is diclizumab, which is another monoclonal antibody that targets a receptor called CD25, which is the alpha subunit of the IL-2 receptor. And it both has some anti-inflammatory effects, but also expands a population of cells called CD56 bright natural killer cells that have a regulatory function. So it simultaneously reduces harmful inflammation and expands the regulatory function of the immune system. And it's dosed as a subcutaneous shot 4 weeks apart. There's currently a phase 3 study ongoing, and in phase 2 studies, you can see a nice dose effect of reducing enhancing lesions in both low and high dose diclizumab compared to interferon beta. And in a phase 2 study, you can see relapse rate reductions of about 50% in diclizumab. So again, just one more option that seems to be highly efficacious. We don't really have a great sense yet of what the monitoring or the potential side effects are or how this is going to fit in with the other drugs we have available. And then lastly, there's an agent called Quinn Ahmad, which for a while was neck and neck with development with BG12 or dimethyl fumarate and with teriflunomide as yet another oral agent. but due to somewhat disappointing effects on relapse rate reduction, the drug is now under further study to look and see if maybe it has an independent effect on reducing disability accumulation, because its effects on slowing disability progression were greater than expected, and they're hoping, I think Teva is hoping for an avenue to use this drug maybe as an add-on agent, as a neuroprotectant, as opposed to a primary reducer of annualized relapse rates. And so what do you do with this sort of vast amount of information and these vast numbers of new therapies? Well, I found it helpful to try and think about these drugs in terms of some of their salient parameters. So if you just plot not all, but most of the drugs that are available, and some of the ones coming available, on a plot where you have safety on one side and efficacy on the other, you can sort of approximate where these drugs may stand in relationship to each other. Now, this is not done using any scientific method. This is just based on published reports of efficacy as well as personal experience. And you can think about some of the older drugs like letermiracetate and the beta interferons having more modest efficacy but doing really well with their safety. Whereas down at the other extreme, you have very effective drugs like Alemtuzumab, natalizumab, that do a great job at controlling the disease, but of course their use is limited by greater risk or less safety compared to the older drugs. And so if you sort of divide this graph up into quadrants, you can see that the one area where we really don't have any treatments is up in this ideal upper quadrant, where we have drugs with superior efficacy as well as very good safety. And so one of the things that I think is incumbent on us as clinicians, as well as the pharmaceutical company that's developing these drugs, is to find ways to either develop new drugs that can fit here, or to stratify the risk for some of these older drugs so that we can select the patients who would most benefit, or for whom it's safe to treat, and sort of move certain patients into this ideal therapy quadrant. So some of the factors, but this is not an exhaustive list, to take into consideration would include considerations of efficacy versus effectiveness. And what I mean by that is that when you read a paper about a clinical trial, you get a sense of what the efficacy of these drugs are in an idealized circumstance. These are highly selected patients. They have very specific disease characteristics, relapse rates, disability levels, and so on. But in practice, the patients that we put on these drugs frequently do not conform to the inclusion and exclusion criteria of a clinical trial. And so when you take all of these other factors into consideration, you get a little bit different picture, and that's what I call effectiveness. So if you take a drug that's approved and you use it in clinical practice, your experience and the benefit those patients receive is going to be a little bit different than what's observed in the clinical trial. And so it's important to take that into consideration. Comparative efficacy is a big gap in what we don't have with MS Therapeutics. There's so many drugs that are so fairly recent, and they're all very expensive and still, proprietary, and that private industry is profiting from these drugs, that we really have very little to go on in terms of comparing one drug against another. Some of the points I'll touch on a little bit more are how we think about treatment of certain patients, at certain points in their disease, or in the sequence that we administer drugs. So some of the newer drugs like Alemtuzumab or ocrelizumab may have long-lasting, if not permanent, effects on a person's immune system. And if we're talking about using a drug like that and then later wanting to use a drug like natalizumab that we know has a greater risk of PML in patients who've been on immune suppressants, then we have to think carefully about the sequence that we use these drugs. Likewise, patients who have very aggressive disease early on, we may need to treat them very aggressively early on, but how do you decide which patients those are, and how do you basically prognosticate at the earliest phases of the disease? Risk stratification is another hot topic. Many of you are probably familiar with the Jc Virus Antibody assay that's specifically developed to help identify patients at higher and lower risk of PML who are going on natalizumab. patients as well as clinicians are interested in dosing, oral versus IV versus subcutaneous shots, as well as some of the safety profiles and adverse events that these drugs carry with them. Dimethyl fumarate is a good example of a drug that appears to be fairly effective, but a lot of patients can be put off by the initial side effects, and so you have to be able to coach patients into what to do to get over the side effects so that they can hopefully stay on the drug long term. Cost, as I mentioned at the beginning, is becoming an increasingly important factor in these drugs, and that many insurance plans are now beginning to limit which drugs are on their formulary and putting in places things like step edits and prior authorization requirements that make it difficult, if not impossible, to select from all of the available agents that we have. And so I'll give you just a couple examples on how I take into account these issues. So in terms of windows of opportunity, I promised you a CAMPATH slide, so at least one of them showed up here. So this is from an earlier phase 2 study that looked at treatment of 2 distinct groups of multiple sclerosis patients. One group had secondary progressive disease, so these are patients who have some fixed disability and are experiencing some slow but continual accumulation of impairment over time. And the other group were patients with early relapsing remitting multiple sclerosis who were much earlier in the disease phase than the patients in the progressive group. And these patients were followed out as long as 3 years in the relapsing group and as much as 10 years in the progressive group. And in the progressive patients, you can see that for the most part, with maybe one exception, eventually everybody still begins to get worse. So there's really no improvement, and the drug doesn't prevent this worsening disability over time. However, if you look at the relapsing remitting group, the patients with the earliest phase of disease, you can see that most of the patients not only stabilize, but they actually improve in their disability very rapidly once the drug is administered, and that for the most part, those benefits are fairly sustained. So this may be a drug that, although it has a somewhat concerning side effect profile, may actually put the disease in long-term or maybe even permanent remission if dosed at the appropriate time in the person's disease. Now, how do you decide who's appropriate for this treatment? I don't know, but that's one of the factors that will need to be taken into consideration in the future. I also included a couple slides on the Jc Virus Antibody because I think it's really taken the drug natalizumab and caused us to approach it using it in a different way. So for those of you who are not familiar with this assay, this is a free blood test that can be obtained on anybody who is either on natalizumab or is thinking about natalizumab or who read about it one time and has no plans at all to go on it. You can order it from any lab, and it can be sent to a central processing facility, and the expense is totally paid for by the company. And the reasoning behind it is that patients who test positive for anti-Jc virus antibodies can be stratified on their risk of developing PML. So if you test negative, your risk basically hangs around here at less than 1 in 10,000, or essentially negligible for as long as you stay on the drug. But if you test positive, depending on whether or not you've been exposed to prior immunosuppressants, you can basically chart a person's risk of PML over time. So in the first 24 months of therapy, the risk of PML, regardless of whether or not you've been exposed to an immune suppressant, is fairly low. But once you exceed that 2-year mark, you can see that the rates of PML jump up substantially, more so if you've been on prior immunosuppressants. So patients in this category who maybe got mitoxantrone at one point or who've been on Imuran at some point, when they hit that 2-year mark, most of the time they're going to come off of the drug and switch on to something else that doesn't have the PML risk with it. More recently, there's been some improvement on the assay, and so if you order the test now, in addition to a positive and a negative result, you'll also get a titer of how reactive a person is to the anti-Jc virus antibody. And it appears that the higher your titer, the greater the risk of PML. So these are a couple of the factors that Dr. Bashir and I and Dr. Metter, our fellow, I know, are using quite a bit in helping patients to decide whether or not this is an appropriate treatment for them and determining how long to keep them on this treatment. And so if you come back to our graph again and you say, well, how do we integrate this new information into the sort of schematic of deciding on treatments, you can see maybe for some patients at least we can move natalizumab up into the ideal therapy category. And so very briefly, I just wanted to mention what's coming down the road. Most of the drugs, as you know, or all the drugs that we have now are basically immune modifiers, and that seems to do a good job at controlling relapses, but we've not really been able to put a dent in this slow progression or accumulation of disability that we call MS progression. And so there's a couple studies ongoing, or about to start or ongoing, that we're involved in. One is supported by Neuronext, which is a new NIH initiative to promote rapid development of clinical trials for neurological diseases. And we're going to be a site for this compound called ibutylast, which is a phosphodiesterase inhibitor. And in phase 2 studies, it showed that patients who were exposed to ibutylast had decreased rates of brain atrophy over time. And so this may be a drug that does not work on the immune system per se, but has a primary neuroprotective effect. And then lastly, I've been working on a project at the VA in which I'm studying whether or not lithium carbonate may have some neuroprotective effects as well. I did a retrospective study to sort of show that patients with lithium exposure who also had an MS diagnosis tended to be stable over time, so the drug did not accelerate their disease. And then I'm doing a pilot study now, a crossover study, in which patients take lithium for either year 1 or year 2 of a 2-year study. And in the end, I'm hoping to be able to show, one, that the drug is safe and well tolerated, which it seems to be thus far, and secondly, possibly get some signal as to what the effects on brain atrophy might be over time. I've currently got, I've recruited 24 patients, all of them progressive, on an assortment of disease-modifying drugs, and adding up my total lithium exposure out through October, got 116 months of on lithium exposure with, I think, 3 discontinuations due to side effects. So the drug, lithium, as you know, has a lot of neurological toxicity, but it seems to be well tolerated at low dosages in these patients. So in conclusion, immune-modifying drugs remain the mainstay of treatment in multiple sclerosis. We do have more and more choices with greater ranges of efficacy and risk, which both make the disease more treatable but also more challenging to treat. Hopefully risk stratification will give us some better idea of which treatments are appropriate for which patients. And in the future, we're looking forward to drugs that maybe move beyond the immune modulating effects and have a primary neuroprotective benefit for our patients. Thank you.